Okay, for this lecture, we're going to be talking about the visual system. This includes the anatomy and physiology of the eye and how we transduce light into brain signals. So first, the eye. This is a fluid-filled sphere, and it has three layers of tissue. So the innermost layer here is the retina. So this is the area that contains all of the neural cells of the eye. So this is the innermost layer of the sphere. Then you have the uveal tract. This is three distinct but continuous structures. So you've got the middle layer, which is called the choroid. The choroid here is rich in capillaries, so it contains the blood that nourishes the photoreceptors in the retina. This contains melanin in the pigment epithelium, and we'll talk more about that later on. There's also the ciliary ring, or sorry, the ciliary body. This is a ring of tissue that encircles the lens. So you have to remember that this is a cross section of a sphere. And so this ciliary body and ciliary muscle, that completely encloses or surrounds the lens. So this is a muscular component, or it has a muscular component that adjusts the lens's refractive power. So it can flatten or cause the flattening or rounding of the lens through a process called accommodation, which we'll talk about in a little bit. And the vascular component of the ciliary body produces the aqueous humor. So the aqueous humor, we'll talk about that in a second, actually. So you've also got the iris here. This is the most anterior component, so toward the front. This is colored and contains two sets of muscles with opposing actions. And so the iris adjusts the size of the pupil, so the space through which light can flow and flow through the lens. And the ciliary muscle is what causes the lens to either flatten or round through that accommodation. And then finally, you've got the sclera. So this is the outermost part of the eye. It's the white of the eye, and it's a continuous bit of tissue that is opaque at the transparent at the front of the eye. So in that part of it's called the cornea. So this is where light comes through the cornea, through the aqueous humor here, and then through the lens, and then through the vitreous humor, and then it is received by neurons in the retina. So the eye has two separate fluid environments, because I mentioned the aqueous humor and the vitreous humor. The aqueous humor is in the anterior chamber. This is a clear liquid. This nourishes the cornea here and the lens. It's produced by the ciliary processes, and it flows into the anterior chamber through the pupil. So this is replaced about 12 times a day. It's drained by a network or meshwork of cells at the junction of the iris and the cornea, and failure of the drainage leads to glaucoma. So if you have too much pressure inside the aqueous body in the anterior chamber, then that can lead to glaucoma. The posterior chamber has the vitreous humor. This is between the back of the lens and the surface of the retina. And this is a clear gelatinous substance. And this is about 80% of the volume of the eye. This holds the shape of the eye. And phagocytic cells clear the debris. So I've mentioned accommodation. This is what allows the eye to focus on near or far objects. So the lens accommodates in order to focus. So for focusing on near objects, that involves a contraction of the ciliary muscle. That contraction of the ciliary muscle reduces tension on the zonal fibers, and that allows the elasticity of the lens to increase its curvature. So if you tighten up this muscle, these fibers become more slack and the lens can bounce back to a rounder shape. So if you have a thin lens, you can see distant objects. If you have a thick lens, you can see near objects. So the shape of the lens itself is determined by two different forces, one of which is the elasticity of the lens. So that tends to keep it rounded up. So the more elastic the lens, the better it's able to round itself when the slack is released from these zonal fibers. And then if there's tension exerted by the zonal fibers, that tends to flatten the lens. So when you're viewing a distant object, the force exerted by the zonal fibers is greater than the elasticity of the lens. And so the flatter shape of the lens is appropriate for distance viewing. But when viewing the closer fibers, if you contract the ciliary muscle, that relaxes the tension on the zonal fibers, and that allows the inherent elasticity of the lens to increase its curvature. Now, that inherent elasticity doesn't last forever. So as you age, the ability of the lens to round up, so accommodate, that decreases. And so this is why people like me need reading glasses. So the lens becomes less elastic with time, and so accommodation isn't quite as effective because that lens can't round itself very well. And so it's harder to see and focus on nearer objects. So on the y-axis here, this is the amplitude of accommodation. So think of it as, this is high accommodation, this is low accommodation, and this is age across time. And as you get older, the capacity for accommodation by the lens decreases. Right, so more terminology. The fundus is the surface of the retina, and there are a few spots within the fundus you should be aware of. The first is the optic disc. So this is a whitish circular area on the retina at the back of the eye. It's the exit point for the retinal axons. And these axons target the thalamus and the midbrain via the optic nerve. It's also the site where the ophthalmic artery enters and the ophthalmic vein leaves the eye. So this is, when we talk about a blind spot, this is a blind spot. So there are no receptors in this particular spot because this is where the optic nerve begins. Then you've got the macula lutea. You'll see it better on the next slide, but it's a circular region with yellow pigment. It's about 3 millimeters right in the center of the retina, and it's an area of high visual acuity. And within that is the fovea. So the fovea is a small depression or pit in the retina, and this region has the greatest ability to resolve fine details. So it has very high acuity compared to the rest of the retina. And so that's why the objects that you're directly looking at appear very sharp and clear, where at times in the periphery, it's more blurry, it's less focused. So this is what you would see when you look through an ophthalmoscope. So the optic disc is over here. It's that white spot. It's the region where the ganglion cell axons leave the retina uh, and form the optic nerve. And the macula lutea here. So this, this kind of bigger area right in the center. And then the fovea is this little depression or pit. So the macula lutea is about 3 millimeters across. So it's the region of the retina that has the highest visual acuity. The fovea is this depression or pit. It's about 1.5 millimeters across, and that lies directly in the center of the macula lutea. So here is a video.
The eye is a fluid-filled sphere enclosed by 3 layers of tissue. Only the innermost layer of the eye, the retina, contains neurons that are sensitive to light and are capable of transmitting visual signals to central targets. These signals are transmitted to the brain via the optic nerve. The immediately adjacent layer of tissue includes 3 distinct but continuous structures, collectively referred to as the uveal tract. The largest component of the uveal tract is the choroid, which is composed of a rich capillary bed important for nourishing the photoreceptors of the retina, as well as a high concentration of the light-absorbing pigment melanin. Extending from the choroid near the front of the eye is the ciliary body, a ring of tissue that encircles the lens and consists of a muscular component that is important for adjusting the refractive power of the lens. The most anterior component of the uveal tract is the iris, the colored portion of the eye. It contains 2 sets of muscles with opposing actions, which allow the size of the pupil, the opening in its center, to be adjusted under neural control. The sclera forms the outermost tissue layer of the eye and is composed of a tough white fibrous tissue. At the front of the eye, however, this opaque outer layer is transformed into the cornea, a specialized transparent tissue that permits light rays to enter the eye. Beyond the cornea, light rays pass through 2 distinct fluid environments before striking the retina. In the anterior chamber, just behind the cornea and in front of the lens, lies aqueous humor, a clear, watery liquid that supplies nutrients to both of these structures. The space between the back of the lens and the surface of the retina is filled with a thick gelatinous substance called the vitreous humor. In addition to maintaining the shape of the eye, the vitreous humor contains phagocytic cells that remove blood and other debris that might otherwise interfere with light transmission. Beyond efficiently transmitting light energy, the primary function of the optical components of the eye is to achieve a focused image on the surface of the retina. The cornea and the lens are primarily responsible for the refraction or bending of light that is necessary for the formation of focused images on the photoreceptors of the retina. When viewing distant objects, the lens is made relatively thin and flat and has the least refractive power. When focusing on a nearby object, the lens becomes thicker and rounder and has the most refractive power. These changes result from the activity of the ciliary muscles that surround the lens. Dynamic changes in the refractive power of the lens to form a sharp image on the retina are referred to as accommodation. Using an ophthalmoscope, the inner surface of the retina, or fundus, can be visualized through the pupil. Numerous blood vessels, both arteries and veins, fan out over the inner surface of the retina. These blood vessels arise from the ophthalmic artery and vein, which enter the eye through a whitish circular area known as the optic disc or optic papilla. The optic disc is also the site where retinal axons leave the eye and travel through the optic nerve to reach target structures in the brain. This region of the retina contains no photoreceptors, and because it is insensitive to light, produces the perceptual phenomena known as the blind spot. Another prominent feature of the fundus is the macula lutea, an oval spot roughly 1.5 millimeters in diameter and located near the center of the retina. The macula is the region of the retina that supports high visual acuity. Acuity is greatest at the center of the macula, a small depression or pit in the retina called the fovea. The restriction of highest acuity vision to such a small region of the retina is the main reason humans spend so much time moving their eyes and heads around, in effect, directing the foveas of the two eyes to objects of interest. The rapid orienting movements of the eyes are called saccades.
So if you recall from one of the earliest lectures, there are different types of neurons within the retina that are highly specialized. So including the retinal bipolar cell, the ganglion cell, amacrine cell, there are also photoreceptors and horizontal cells. And we'll talk about those in detail in just a minute. So it's important to note that these photoreceptors here, and we'll talk more about these in a little bit, these rods and cones, they're embedded in the outermost layer of the retina, and they are what's responsible for converting graded electrical activity into action potentials. And then these travel to their central targets via the optic nerve. So the retina has layers to it. So there are cell bodies of 5 different neuron classes that are located in inner nuclear, outer nuclear, and ganglion cell layers. And these combine to process the light. So they do that through the rods and the cones. And these are on the outermost layer of the retina. So light actually has to travel through non-light sensitive cells to reach the outer segments of the photoreceptors. So, and these are embedded in the pigment epithelium that keeps them healthy, and we'll talk about that in a little bit. And then, so those project onto bipolar cells in the outer nuclear area. So nuclear meaning this is where the cell bodies are. And then you've got the outer plexiform layer, which helps with lateral information flow. So this is where you might find horizontal cells and the amacrine cells. And then those connect also to bipolar cells that then connect to synapse onto the ganglion cells, and the ganglion cells are what combine and converge to form the optic nerve. So the path of information signaling goes from the photoreceptor here to the bipolar cell and the ganglion cell, sorry, the bipolar cell and then the ganglion cell. That is the most direct path. But there's also integration of information. So that's where the horizontal cells and the amacrine cells, they provide lateral interactions and help to maintain the visual system sensitivity to contrasting light intensity. So this helps with integrating information across what would otherwise be very direct paths from the photoreceptor to the bipolar cell to the ganglion cell that forms the optic nerve. Now, the photoreceptors, as I mentioned, are embedded in the pigment epithelium. The light absorption initiates an intracellular cascade within this cell, and this changes the membrane potential of the receptor. So light absorption alters the membrane potential. This is phototransduction. So they are, as I've said a few times, embedded in the pigment epithelium. This is the outermost layer of the retina. The light has to travel through non-light sensitive cells in order to reach these photoreceptors. And these epithelial processes here extend down between the outer segments of the photoreceptors and keep them healthy. So the discs that form the photoreceptors only live about 12 days. And there are new outer segment discs continuously being formed near the base, and then the older discs are removed or phagocytosed. So they've measured here, what you can see is they're forming new discs, and those are traveling up, and then these the discs start to curl. So the oldest ones are the ones that are closest to the pigment epithelium, and the tip becomes spherical and starts to separate from the rod, and then the tip is engulfed by the pigment epithelium. Now, phototransduction itself, light activation causes graded changes in membrane potential. So we've talked about this from a few different perspectives now. We've talked about it from synaptic potentials and receptor potentials in response to touch. So this is another kind of potential evoked by lights. So this is also somewhat different because there aren't action potentials. These are very, very small cells. So the changes, the graded changes in membrane potential also correspond to a change in the rate of neurotransmitter release. So if you shine a light on a photoreceptor, whether it's a cone or a rod, that leads to membrane hyperpolarization. So look here at the y-axis with the membrane potential. And so it's resting at about -40 in this case. There's a flash of light, and it causes hyperpolarization, not depolarization. So in the dark, the resting membrane potential is about -40. And if you add light, it kind of dose dependently, and you can't see my little finger quotes right now, hyperpolarizes the membrane. So they did this in a turtle retina, and at varying intensities of light, you get various degrees of hyperpolarization. So the least intense flash response doesn't really hyperpolarize the cell very much. The most intense flash of light hyperpolarizes the cell to about -65. So this, it doesn't seem intuitive given everything else that we've talked about in the course so far, but and here's a quote from the text. The only logical requirement for subsequent visual processing is a consistent relationship between luminescence changes and the rate of transmitter release from the photoreceptor terminals. Just because it happens to be in the opposite way that we would anticipate, it doesn't matter. So more light means hyperpolarization and less neurotransmitter release. So in the dark, the resting membrane potential is about -40. In the dark, the cyclic GMP levels in the outer segment membrane, these are high. So the cyclic GMP binds to the sodium permeable channels, keeping them open and allowing sodium and other cations to enter, so causing the cell to be depolarized. So that's the steady state in the dark. You have depolarization to that -40. But in the light, absorption of the photons leads to a decrease in cyclic GMP levels, and thereby closing the cation channels, resulting in hyperpolarization. So in the light causes the cell to become hyperpolarized. And I'm really trying to emphasize this because it's not, because it isn't intuitive. You would think that light would cause the cells to do something, when in fact, it causes the cell to stop doing something. So whether it's more or less, or on or off, or stimulus or no stimulus, as long as it's consistent and that signal is being consistently received and consistently sent, it's just the way it is. So light causes hyperpolarization of photoreceptors. Yeah, and regardless of the seeming direction of polarization or depolarization, there's a stimulus-specific graded change in how much neurotransmitter is being released. Here's a video.
The retina contains two types of photoreceptors, rods and cones. Both types have an outer segment composed of membranous discs containing light-sensitive photopigments. Each also has an inner segment that contains the cell nucleus and gives rise to synaptic terminals that contact bipolar and horizontal cells. Phototransduction is the process of a cell absorbing light and creating a response. The response is a change in the amount of transmitter that is being released onto target neurons. The photopigments lie within the disc membranes of the outer segments. In rods, the photopigment is called rhodopsin. The 7 transmembrane domains of the protein portion of the molecule, called opsin, traverse the membrane bilayer. The opsin forms a pocket in which the light-absorbing portion of the photopigment, a molecule called retinal, resides. The photopigments in the rods and the 3 types of cones differ in the protein portion of the molecule, which tunes retinal to absorb specific wavelengths of light. Here, retinal is in a conformation called 11-cis, with cis referring to 2 hydrogen atoms being on the same side of an important double bond. When retinal absorbs a photon of light, a component of this double bond breaks, allowing free rotation about the bond. This change in retinal to an all-trans form triggers a series of alterations in the opsin component of the molecule. The changes in rhodopsin lead to a cascade of events in the cell. The next component in the cascade is a trimeric G protein called transducin, which in its inactive state is bound to GDP. The altered rhodopsin molecule activates transducin, allowing it to exchange its GDP for GTP. The alpha subunit of transducin then activates a phosphodiesterase in the disc membrane. Phosphodiesterase hydrolyzes cyclic GMP, thus lowering the concentration of cyclic GMP throughout the outer segment. As the concentration of cyclic GMP falls, this molecule no longer binds to and holds open ion channels in the surface of the outer segment membrane. Let's compare the action of the ion channels in the light and in the dark. In the dark, the ion channels are open. The influx of the positively charged sodium and calcium ions has a depolarizing effect on the cell. The inward current is opposed by positively charged potassium ions flowing out through potassium channels, a hyperpolarizing influence. The combined result of these ion flows, however, is the depolarization of photoreceptors in the dark. In the dark, the depolarized state of the membrane, which is at approximately -40 millivolts, triggers continual transmitter release from the synaptic terminals of the photoreceptor cells. Absorption of light reduces the concentration of cyclic GMP in the outer segment, leading to the closure of the cyclic GMP-gated channels. As a result, positive charge carried by potassium ions flows out of the cell more rapidly than positive charge carried by sodium and calcium ions flows in. The cell becomes hyperpolarized, that is, more negative, and decreases its release of transmitter. One of the important features of this cascade is that it provides enormous signal amplification. A single light-activated rhodopsin molecule can activate an estimated 800 transducin molecules. Although each transducin activates only one phosphodiesterase molecule, each phosphodiesterase is capable of breaking down as many as 6 cyclic GMP molecules, resulting in the closure of approximately 200 ion channels. This number of channels represents 2% of the number of channels in each rod that are open in the dark, and their closure causes a net change in the membrane potential of about 1 millivolt. Proteins in the photoreceptor limit the duration of this amplifying cascade and restore the various molecules to their inactivated states. Activated rhodopsin is rapidly phosphorylated by rhodopsin kinase, which permits the protein arrestin to bind to rhodopsin. Bound arrestin blocks the ability of activated rhodopsin to activate transducin, thus effectively truncating the phototransduction cascade. By hydrolyzing GTP, activated transducin has a built-in timing mechanism to turn itself off shortly after becoming activated. Without activated transducin, phosphodiesterase also turns off. Finally, through a multi-step pathway, the cell converts rhodopsin back to a form that can absorb light. Also in the dark, The enzyme guanylate cyclase again builds up the levels of cyclic GMP. Cyclic GMP opens the ion channels, allowing an influx of ions and depolarizing the cell.
So after the photoreceptors have transduced the light signal and the path through the bipolar cells and then the ganglion cells has been activated, these signals need to get sent to the brain for further processing. So the optic nerve ganglion cells, they leave the eye through the optic disc and form cranial nerve 2. Uh, and then at the optic chiasm, so the cross here, about 60% of fibers cross to the contralateral side, and 40% continue ipsilaterally to the thalamus and midbrain targets. So about 60% of fibers leaving the eye cross at the optic chiasm and innervate contralateral brain areas, so the other side of the brain. Let's see. So along the optic tract, so these contain ganglion cells and fibers from both eyes. So we've got 40% remaining ipsilaterally, 60% crossing contralaterally. This happens from both eyes. So from this eye, you've got 60% of its fibers joining up with the 40% of the ipsilateral fibers from this eye. So the optic tract contains ganglion cell fibers from both eyes, and its major target is this lateral geniculate nucleus of the thalamus. So axons from the lateral geniculate nucleus then, these project to the striate cortex here. So this is inside the optical lobe, the optical cortex, and this is part of the primary visual cortex, so V1. So this is the optic radiation that leaves the geniculate nucleus of the thalamus and projects to the optic cortex in the primary visual cortex. So the second major target of these ganglion cells is the pretectum. So the pretectum is responsible for the pupillary light reflex. So this interacts with circuitry of the oculomotor nerve. And then there are other projections and targets of the optic nerve. So the suprachiasmatic nucleus here of the hypothalamus. So this is involved in circadian rhythms, so maintaining a day-night cycle, so a sleep cycle. And then the superior colliculus. Oh, sorry, that was the that was the hypothalamus. So the suprachiasmatic nucleus. Suprachiasmatic, so above the optic chiasm. So supra above chiasmatic, the chiasm here, and it's part of the hypothalamus. So that is involved in the circadian rhythms, and then the superior colliculus coordinates head and eye to visual targets. So these projections involve ganglion cell types that are distinct from cone and rod light sensitivity. So these involve a different system than the cones and the rods. And yeah, so kind of a third light sensitivity system, and that's still being elucidated. If you want to look into it, it has to do with the detection of blue light, and that is very much involved in that circadian rhythm maintenance. So this was kind of shown a little bit in that first video that you watched, but just to make it really very clear, the passage of light rays through the pupil of the eye actually results in images that are inverted and left-right reversed on the retinal surface. So what we believe that we're seeing isn't what's being reflected on the retina. So each eye sees part of its visual space that defines its visual field. So here we've got the left visual field. This is what your left eye can see. And then this is your right visual field. This is what your right eye can see. The portion that does not overlap in each of these visual fields is known as the monocular portion of the visual field, so single eye. And the portions where these visual fields do overlap is known as the binocular visual field. So this is, again, the left monocular visual field here and the right monocular visual field here. You've got the nasal division, so toward the nose, and you've got this blind spot here, not the same as the optic disc blind spot, but the nose kind of impedes what the visual field might have been otherwise. And then you've got the temporal side here. So if you think about the temporal lobe being most lateral part of the brain, this is the temporal portion or division of the visual field. And then you've got superior, so above, and inferior, so below. Now, on the retina, and that's what these images are down here, the visual field is inverted, and so upside down, and left, right, reversed. And so here's a better image of what's being perceived here. So where the left-right visual fields overlap, that's the binocular vision, and each of these is being projected onto the eye and then across. So it enters through the cornea, goes through the aqueous humor, the pupil, the lens, vitreous humor, and then hits the retina. So the light hits the retina and it hits those photoreceptors. And you can imagine, so this, for instance, the far left visual field here, point A, is being perceived by the eye. So this is the far left being perceived, and then it's actually hitting the retina on the far right side. So and for instance here, section B, so a point of binocular vision. So section B is being perceived by both eyes. And again, it's slightly to the left, but it's hitting slightly to the right on the retina. So that's how the visual field is reflected on the retina itself. So as it says here, regardless of whether it originates from the left eye or the right eye, the left half of the visual world, so this left visual field, is represented in the right half of the brain, and the right half of the visual world is represented in the left half of the brain. And a video.
The human eye has many of the features of a camera, starting with a lens to focus light. The cornea of the eye bends light rays and is primarily responsible for forming the image on the retina, the receptive surface inside the back of the eye. The visual image focused on the retina is inverted top to bottom and reversed right to left. The part of the world that you can see without moving your head or eyes is called your visual field. Each eye sees only a portion of this visual field. The visual field can be divided into right and left visual hemifields. The right visual hemifield is seen by the temporal left retina and nasal right retina, while the left visual hemifield is seen by the nasal left retina and the temporal right retina. The visual field can also be divided vertically into superior and inferior divisions. Note that the visual fields of both eyes overlap extensively in the central portion of each visual hemifield. This region defines the binocular field of view. Vision in the periphery of the field of view is strictly monocular, mediated by the most medial portion of the nasal retina. The center of the visual field projects its image onto the foveal region of the retina. On the retina, the highly specialized fovea has a dense concentration of small diameter cones and a one-to-one relationship between the cones and the bipolar and ganglion cells. Visual acuity is therefore especially high in this region. Now let's examine the pathways from the retina into the brain. This diagram shows a view of the base of the brain as seen from below. Note that in this view, the position of the two eyes is reversed. The axons of retinal ganglion cells exit the retina via the optic nerve. The optic nerve exits the eye in a region called the optic disc. Because there are no receptors in this region, nothing can be seen in the corresponding part of the visual field. This blind spot does not appear as a dark spot. Rather, it is simply a region from which we cannot obtain visual information. The optic nerves cross at the optic chiasm, which is located just anterior to the stalk of the pituitary gland. In humans, axons from the nasal retina cross over to the opposite side of the brain. Axons from the temporal retina project to their own side of the brain. After they pass the optic chiasm, the axons of the retinal ganglion cells are known collectively as the optic tract. Note that information from the left visual field is carried in the right optic tract, and information from the right visual field is carried in the left optic tract. The vast majority of axons of the optic tract terminate in the lateral geniculate nucleus, or LGN, which is the visual part of the thalamus. However, the axons of retinal ganglion cells also project to several other brain regions. Some axons of retinal ganglion cells extend to the superior colliculi, a paired structure on the roof of the midbrain. The superior colliculi help coordinate rapid movements of the eyes toward a target. Small bundles of optic tract axons also project to the suprachiasmatic nucleus in the hypothalamus. Cells in the suprachiasmatic nucleus are involved in the control of daily or circadian behavioral rhythms related to the light-dark cycle. Finally, optic tract axons from still other ganglion cells project to a collection of neurons that lies between the thalamus and the midbrain in a region known as the pretectum. This region is important as the coordinating center for the pupillary light reflex, that is, the reduction in the diameter of the pupil that occurs when sufficient light falls on the retina. The lateral geniculate nucleus serves as the primary relay nucleus for visual processing by the cerebral cortex. The right lateral geniculate receives information from the left visual field, the nasal left retina, and temporal right retina, while the left lateral geniculate receives information from the right visual field, the nasal right retina, and the temporal left retina. The spatial relationships among the ganglion cells in the retina are maintained in their targets as orderly representations or maps of visual space. The primate lateral geniculate nucleus has 6 layers, and inputs from the two eyes are maintained in separate layers within the LGN. From the lateral geniculate nucleus, visual information is relayed to the visual cortex. Most of the axons from LGN neurons form the optic radiations, which terminate in the visual areas in the occipital cortex at the back of the brain. In this lateral view of the brain, we see that axons carrying information about the superior portion of the visual field sweep around the lateral horn of the ventricle in the temporal lobe, a branch called Meyer's loop, before reaching the occipital lobe. Axons carrying information about the inferior portion of the visual field travel in the parietal lobe. The topographic order of visual information is maintained in the visual cortex. The fovea is represented in the posterior part of the visual cortex, whereas more peripheral regions of the retina are represented in progressively more anterior regions. Note that the area of central vision, the fovea, is represented over an especially large part of the visual cortex. Inputs from the two eyes converge at the cortical level, making binocular effects possible. The primary visual cortex, V1 or striate cortex, projects to other areas of the cerebral cortex, referred to as extrastriate, that are involved in complex visual perception. The dorsal stream, which includes the middle temporal area, leads from striate cortex into the parietal lobe. This system is thought to be responsible for spatial aspects of vision, such as the analysis of motion, and positional relationships between objects in the visual scene. Another system, the ventral stream, leads from the striate cortex into the inferior part of the temporal lobe. This system is thought to be responsible for high-resolution form vision and object recognition.